In 1986, the world witnessed one of the worst nuclear disasters in history when reactor number 4 at the Chernobyl nuclear power plant exploded. The explosion released massive amounts of radiation, creating a 30-kilometer exclusion zone that remains highly radioactive to this day. Despite the devastation, life has found a way to persist in this inhospitable environment. A black mold called Cladosporium sphaerospermum was one such organism. It was found growing on the walls of the destroyed reactor. This mold is radiotrophic, which means unlike typical molds, this fungus thrives in the extreme radioactive environment of the exclusion zone. Using melanin in its cell walls, this fungus converts gamma radiation into chemical energy through a process called radiosynthesis, analogous to how plants use sunlight for photosynthesis. First observed in 1991, the mold was found to be growing towards radioactive sources, a behavior termed radiotropism, and can withstand radiation levels lethal to most life forms. This fungus has sparked scientific interest for its potential in cleaning up contaminated environments, as it may help reduce radiation levels in contaminated areas like Chernobyl or Fukushima by absorbing and neutralizing radiation. In one experiment on the International Space Station back in 2016, showed that a 1.7 mm layer of Cladosporium sphaerospermum reduced radiation by about 2%, suggesting applications for protecting astronauts from cosmic radiation during long space missions, especially when looking at sending astronauts to Mars. An estimated 21 centimeters thick layer of the mold would be needed to reduce the annual dose of radiation on the surface of the red planet. Over 200 fungal species have been identified in Chernobyl, with melanin-rich ones like Cryptococcus neoformans and Weighingella dermatitidis also showing radiotrophic traits. The exact mechanism of radiosynthesis remains under study, but melanin's ability to absorb and convert radiation is key. The Chernobyl disaster, despite its devastating impact, has inadvertently provided a unique environment for studying the resilience of life. The discovery of Cladosporium sphaerospermum and its radiation-eating properties is a testament to the adaptability of life in the face of extreme conditions. It reminds us that even in the most inhospitable places, life finds a way to thrive.